This is my presentation on chloride ions in the central nervous system and skeletal muscles. GABA or gamma-aminobutyric acid is the main inhibitory neurotransmitter in the brain. It controls feelings of fear and anxiety. This neurotransmitter hyperpolarizes its target cells by opening chloride ion channels and allowing the chloride ions to enter the cell. The hyperpolarizing of the cell makes it less likely for the cell to fire action potentials, which then reduces the excitability of the neurons. There are two types of GABA receptors we will look at. GABA A receptors are ligand-gated chloride ion channels that open to allow the influx of chloride into the cell. This causes the cell to become more negative. Therefore, the activation of GABA A receptors increase the intraneural concentration of chloride ion, hyperpolarizing the cell. GABA B receptor sites, on the other hand, are heterodimers coupled to G proteins. The main response to GABA B receptors activation is an increase in potassium conductance, which also causes the hyperpolarization of the neuron. The GABA B receptors also reduce the neural calcium conductance, which is thought to be responsible for baclofen-induced inhibition of neurotransmitter release. These actions reduce the excitability of neurons in the central nervous system and leave a calming effect on the brain, also reducing the feelings of fear and anxiety. Two drugs that work due to their effects on GABA A receptors and chloride ions are benzodiazepine and barbituric. Benzodiazepine is often prescribed to patients with anxiety and panic disorders due to its effect of reducing activity of nerves in the brain. The mechanisms of action of benzodiazepine agonist is to enhance the GABA transmission. Benzodiazepine increases the frequency of GABA A ligand-gated chloride ion channels opening in response to GABA. Similarly, barbiturates reduce activities of nerves and cause muscle relaxation. They increase the binding of benzodiazepine and GABA to their respective binding sites. They also increase the GABA A channel open time, resulting in an overall increase in mean channel open time and chloride ion flux. Both barbiturates and benzodiazepine have inhibitory effects on the central nervous system and can decrease the activity of excitatory neurotransmitters and work in synergy. Having a combination of barbiturates and benzodiazepine can lead to profound nervous system or respiratory depression, which may lead to death due to breathing that is inadequate for supplying oxygen to the body. Chloride ion channels participate in many important physiological tasks in the human body. The chloride ion channel, CLC1, specifically is critical for the function of skeletal muscles. This channel is a voltage-gated channel located at the T tubules and sarcolemma in the skeletal muscle. Its main physiological function is setting and restoring the membrane potential of skeletal muscle because of chloride ions' negative charge. The CLC1 channel physiological roles in the skeletal muscles more specifically include the activation of chloride ion channels to ensure the electrical stability of skeletal muscle by resetting its membrane excitability to the resting state after firing an action potential, and maintaining an effective chloride ion homeostasis system in order to help in the generation and propagation of action potentials. Myotonia congenita is a channel channelopathy that affects chloride conduction in the skeletal muscle. It is a genetic muscle disease caused by mutations in the skeletal muscle. CLC1 gene on the chromosome 7. This mutation in the chloride ion channel leads to the absence of the stabilizing influence of the chloride ion fluxes and results in the hyperexcitability of the plasma membrane of skeletal muscle fibers. It is due to an electrical instability of muscle membrane itself and leads to the repetitive action potentials with a single stimulus. It can be inherited in an autosomal recessive Becker type or dominant Thompson type. It is characterized by the inability of the skeletal muscles to quickly relax after voluntary movements. Symptoms begin early on and can change from person to person. They may include muscle stiffness, muscle weakness, and attacks of weakness brought on by movement after rest.